Hello and welcome back to Science. Today I'm going to be talking about how vesicle release occurs at the synapse. So if you're familiar with how a synapse works, what normally happens is you have an action potential coming down your axon here and it reaches your synaptic node. So I've just drawn out a very basic presynaptic node here and a postsynaptic node at the bottom here. And what usually happens is you have calcium coming into your presynaptic node, which causes the release of vesicles, which are little pockets almost, which contain neurotransmitters, which can diffuse across your synapse and activate little receptors like this one here on the postsynaptic membrane, hence transmitting your signal between different neurons in a chain. And so today I want to explore exactly how we go from the action potential arriving at this scene to the release of that neurotransmitter into the synapse. That is the key point that we are going to be focusing on. I will probably focus on other parts of the process in other videos. So if you're interested in that as well, make sure to subscribe. But by getting straight into it, we have to sort of think about where these vesicles actually come from, because long before we have the action potential coming in, we actually have our vesicles already in play. So I'm just going to draw, I have no idea how big to make these. Let's make our little vesicles this size, obviously not quite to scale. I'm going to make vesicles purple just because it stands out a little bit more against the background. So this is my vesicle here. And essentially, if you don't know what a vesicle is, it's basically just a piece of membrane, which is sort of in a sphere. And you can store things inside this little sphere of membrane here. In this case, it's neurotransmitters. And what you can actually have is you have these little transporters here, which are going to actively pump your neurotransmitter into your membrane, into your vesicle, sorry, by active transport. So that's requiring ATP. Your neurotransmitter will generally be synthesized in different parts of the cell depending on what your neurotransmitter is, but it's synthesized outside of the vesicle in a lot of cases, and then it's actively transported in. Pop some ATP here just so it's obvious. And when you have this packing of this neurotransmitter into your vesicle, vesicles are essentially packed in something known as quanta, which essentially means that every vesicle contains the same amount of neurotransmitter. And so these active transporters are very important for ensuring that every vesicle has the exact same quanta of neurotransmitter. And this quanta can be very different depending on the synapse. Some synapses have very small quanta, only vesicles will only have a small number of molecules of neurotransmitter in them. Some synapses will have very large quanta, huge numbers of neurotransmitter in them. It's really dependent on the synapse, on the neurotransmitter, and sort of on the needs. But there is this sort of quantified amount that is released, and these active transporters are very important in making sure that that quanta is met. But at the moment, our vesicle is actually in a sort of resting state. We haven't had any stimulation yet. And this is what we call the reserve pool of vesicles, where they're kind of just chilling. They're not doing anything. They're kind of hanging around waiting for a signal. But if they were just kind of there in a cell like this, they would kind of be a problem. They'd be just floating around in lots of different places. So what you actually have whilst they're in this reserve pool is you actually have them anchored to the cytoskeleton of the cell. The cytoskeleton is basically like our skeleton. It's sort of like a framework made up of different proteins inside the cell, which essentially allow the cell to keep its shape and keep certain structures in place. So in this case, this is just representing a part of the cytoskeleton here, but this would usually, in the case of vesicles, be a protein in the cytoskeleton known as actin. And essentially between the vesicle and the actin here, we have another protein. There's going to be a lot of proteins today, but we're going to have this little guy in here. I'm going to try and do all the proteins different colors. I don't know, I might run out of colors. Let's make this one red. So this protein is known as synapsin, and it is going to play a really big role in when our neurotransmitter filled vesicles are in their reserve pool compared to when they are going to actually be coming down to the membrane here to fuse and release their neurotransmitter. And that is essentially where it links into this idea of the action potential. So when you have an action potential coming down your axon here, this is sort of just a little stub of the axon at the very end, you have this ion exchange. And what this is going to cause is these calcium channels at the end of these axon here will be opened by the membrane depolarization, and what that's going to cause is calcium ions to come into this cell. So these are voltage dependent calcium channels. And calcium plays a lot of different roles inside neurons. It's used for membrane depolarization, but it's also used as a very important signaling molecule. And that is exactly what it's going to do now. It actually interacts with several proteins, but most importantly for our purposes here are kinases. Now, kinases are essentially proteins that phosphorylate things. So we have MAP kinase and CAM kinase, which are activated essentially by this calcium influx into the cell. And once these are activated, as kinases do, they like to phosphorylate things. What are they going to phosphorylate? Well, they're going to phosphorylate our friend here, the synapsin. I've just drawn another one here. This is supposed to just be the same thing, but essentially I just wanted to make it closer so I could more easily draw an arrow to it. And I'm just going to represent phosphorylation with this PI here, this inorganic phosphate symbol. But what's phosphorylating this synapsin does here is essentially it causes it to unbind the vesicle from this actin cytoskeleton, this sort of mesh that it's been bound to. And so suddenly our vesicle finds itself free in this synaptic node here. And that means that it can be transported from its humble abode out here in the cell all the way to this membrane. I'm going to make this arrow a little smaller because I now realize I've given myself a lot of space in the middle, which I've not used. So I'm just going to make all this stuff down here really big. Our vesicle is going to be transported all the way over to the membrane here, and it is going to undergo a process known as docking to the membrane. So I'm just going to draw it a bit bigger just so you can sort of see what's going on a bit better, because there's going to be a lot of proteins interacting with the membrane here. So down here, I've just drawn the membrane layers of the presynaptic node. This is just the lipid bilayer here. That's all this is representing. This is the vesicle, which is technically also a bilayer, but I don't want to have to draw two circles every time I draw a vesicle. So, but the membrane of the vesicle is also a phospholipid bilayer. And essentially, when the vesicle comes down to this membrane here, we have several different proteins that we need to think about. The proteins we really need to worry about are proteins involved in constructing something known as the SNARE complex. And the SNARE complex is a combination essentially of this protein, which is called synaptobravin 2. And then we have this protein on the cell membrane, which is called cynotaxin 1. And then that is also associated itself. We have these other two here known as SNAP proteins, SNAP25 proteins to be exact. They all sound very similar and they are all very easy to get muddled, which is why I'm going to try and do them all in different colors so that when I label these, it's really obvious which is which. And these three types of proteins, there's 4 in total, are going to form the SNARE complex, and that is basically going to be driving this whole vesicle fusion with the membrane, the association of these proteins here. So these are mostly alpha helical proteins. These sort of long chainy like bits are essentially really long alpha helix domains, which are sort of like big spirals if you've ever seen them on proteins before. And so they are going to want to associate at some point, but they're going to need some sort of driving force to associate them. So if I sort of just draw on this side here, maybe as they're sort of getting a little bit closer to each other, but what you're now going to get is another protein get involved on the action. Like I said, there's a lot of proteins. This is known as MUNC18, and that is essentially going to act as a zip. So it's sort of going to go along here and essentially zip the alpha coils together. And as it sort of moves along and brings them closer together, what that's going to do is it's going to bring this vesicle closer and closer to the membrane, and eventually it's going to push it into the membrane when we get to that point. But the problem with this is there is another protein. This time, this is one is sort of going to be, I'm trying to draw this in a way you can see everything. I'm just going to pop him kind of here. This one is called complexin, and complexin is essentially going to block this monk protein from zipping the SNARE complex the whole way up. So this is sort of gonna zip sort of halfway or a decent way along, but it can't fully zip the vesicle up. And what that is going to result in is our vesicle here is going to get really close to the membrane, but it's not actually going to be able to fuse. And so what we now have is we have this vesicle here, which has been transported to the membrane, has been docked, and it's what we would call in the primed position, as it's sort of ready and activated. We've not actually had this calcium signal yet to tell us that we need to release our neurotransmitter, uh, but we have had this sort of preparation, and you'll always have a certain number of vesicles which are in this prime position, ready to go at any synapse. As soon as that calcium comes in, they can release their neurotransmitter into the synaptic space, and you can get that really fast response, which is so characteristic of nerve transmission and synapses. So this is sort of how a lot of vesicles in what we call the active zone are sat. So we had the up here with the reserve pool, but down here we have what's known as the active zone. Everything's sort of primed, ready to go, and as soon as we get that calcium influx, everything is going to fuse with the membrane, and we're going to get that neurotransmitter release. But how exactly does that calcium input from our action potential cause this release? So if I just draw us in a little calcium channel here in our membrane, and the calcium channels are located very close to where the synapses are, because calcium is very tightly regulated within cells, especially within neurons, as it is such an important messenger molecule. So as soon as it sort of gets beyond what's known as the calcium microdomain, which is basically means that the immediate area around the channel, it's very quickly removed or buffered by various processes in these cells. So in order for the vesicles to actually feel the effects of the calcium, they have to be very, very close to where it is being pumped into the cells. And so that means they have to be sort of quite close to this calcium microdomain to actually sort of feel the effects of this rapid increase in concentration of calcium. But what does our calcium actually affect? Well, fun fact, it's actually another protein on this SNARE complex here. And I am just pondering what color I could possibly use. Well, you've not used brown yet. Let's use brown. So I'm just going to draw this as another circle, because at the end of the day, this is a very simplified representation. There's loads of different proteins going on here. These are the most important. So that doesn't really stand out, does it, in brown? Orange. It's going to be an orange protein. Anyway, this little orange guy here is going to be the protein known as synaptotagnin. And if you're thinking that the people who named these proteins were really not very creative, you've probably hit the mark there, because they all include some variant of the word synapto or synap, or you can tell where they come from, but it's really easy to get them confused with each other. And essentially, what this calcium is going to do is it's going to interact with the C2 domain on synaptotagnin. C2 is just a domain on synaptotagnin which interacts with the calcium. So this binding of the calcium to the C2 domain of synaptotagnin causes it basically to remove this complexin molecule here. And if you remember, this complexin is essentially what's stabilizing the molecule in this primed state. It's essentially keeping the vesicle like this in this prime state, ready to go. It's preventing MUNC18 from fully zipping up our SNARE complexes here and fully fusing them together. So if we remove this complexin, what's going to happen is our MUNC can fully zip up the SNARE complexes, and the vesicle can fully fuse with the membrane. So what essentially happens is these SNARE complexes here get tighter and tighter as they're zipped closer and closer together, is you get this sort of weird looking state here as I've drawn it, which we call hemifusion, where essentially the outer leaflets of the cell's membrane and the vesicle's membrane fuse. Now this looks really weird because I've drawn my vesicle in a different color from my cell membrane. But you have to remember that this lipid membrane and this lipid membrane are essentially made of the same thing. So this darker yellow and this darker purple are basically the same thing. This sort of lighter yellow here and this lighter purple in here are basically the same thing. So what you're sort of getting with this hemifusion is these outer lipid layers of your phospholipid bilayers are becoming the same things. You still have this distinct package area here on this sort of inner membrane. This is still an enclosed space, but the outer membranes are now interacting with each other. So all of these vesicles in the past should have had this double membrane here. I just haven't been bothered to draw out the phospholipid bilayer with phospholipids on both sides. But I've just been lazy and not drawn both layers there because this is a lot more complicated looking of a thing. And unfortunately, I've not really been able to draw it very well here just because I've colored the vesicle in a different color. It does look a little bit weird. This is sort of like the final step before you get like complete fusion. This monk is sort of fully zipped along. It's gone right from the end all the way along. These snare complexes are getting really tight and really compact. And so eventually what that is going to end up with is a complete fusion with the membrane. I'm just trying to think how I should draw this now because this is also going to look weird because I've used different colors for the vesicle and the membrane. And so as your vesicle fuses with your membrane, as I've tried my best to represent here with, again, like different colors doesn't quite make as much visual sense. This sort of blue space out here and this purple space in here essentially become the same space, and any neurotransmitter you have in your vesicle is going to come out into your synapse and it's going to diffuse down to your receptor here, and it can interact with that receptor and cause an effect. And that is essentially how your vesicle goes from being created, having that neurotransmitter initially pumped into it up here, all the way to releasing that neurotransmitter into the synapse. But that's not necessarily the end of the story in all cases. I mean, this is sort of a fairly case-by-case basis. But a lot of times, especially with neurotransmitters rather than like neuropeptides or something, which you wouldn't necessarily see small synaptic vesicles like these being used for, is you would actually recycle sometimes your neurotransmitter. There are several neurotransmitters which are reuptaken back into the cell, and a lot of these are by endocytosis. So this process of fusion here is something which is called exocytosis. You are merging a vesicle with the membrane to release something outside the cell. Endocytosis is the opposite. You are causing like an invagulation, a sort of indent into the membrane, absorbing something from the outside and taking it into the cell. So this time I've drawn the invagulation into the membrane for the endocytosis in the same colors as the membrane and its sort of extracellular fluid here, but in essence, these pictures should sort of be the same thing. This is the same as the yellow, and this is the same as the purple. And so what you get is your neurotransmitter that you sort of had in your synapse gets taken back up into these vesicles here. When it served its purpose, it stimulated the action potential, and you don't want to keep stimulating action potential because that is ultimately bad. If you get too much stimulation, that can cause you lots of side effects associated with various neurological diseases. So we want to bring our neurotransmitter back into the membrane, and we are going to do that through this endocytosis. And this is essentially going to create for us just a new vesicle. So if I do this is essentially just this bit of the membrane here, which is fully enclosed, and it's sort of wrapped around and pinched off, basically the opposite to what we've just done down here. And what you can do once you have your vesicle full of neurotransmitter again, which you've just taken back in from the membrane, is just bring it straight back to that reserve pool and recycle it. Now, you can't do this in every synapse. Not every neurotransmitter is recycled. Some are broken down by enzymes such as acetylcholine, which is broken down into acetyl and choline, funnily enough. But some are just fully reuptaken into the cell as is. Some are, once they're broken down, like acetylcholine, the different bits of them are taken up like this, and then they're sort of pieced back together again. But essentially, this sort of creates what we call the small synaptic vesicle cycle. As you can see, it is a cycle. They start in the reserve pool, they go down, they dock with the membrane, they release the neurotransmitter. Neurotransmitter gets retaken back up into the cell. That goes back up into the reserve pool and waits for the next stimulation. It goes around and round and round. And these guys are known as small synaptic vesicles. So you do have these other vesicles known as large dense core vesicles, or LDCVs. They are different from small synaptic vesicles because they contain neuropeptides instead of neurotransmitters. Neuropeptides essentially modulate the activity of neurotransmitters at the synapse. They're a slightly different thing. I might do a video on them in the future, so let me know if you're interested in seeing that. But this is just focusing on small synaptic vesicles, or SSVs. And so now you can see how we go from getting this initial calcium influx for one action potential, and then maybe another calcium influx for another action potential. As these guys, one calcium influx might cause these guys to release, and so they need replacing. So that same calcium influx causes these guys to come down and be in the prime position. So the next time you get a calcium influx, they are then ready to go, and you have a readily releasable form of vesicles in the active zone, which I have neglected to label and will now label. And so you always have something you can release. This is not necessarily always the case. Sometimes you have potentiation where you run out of readily releasable vesicles and stuff like that. But that's really a topic for another video. It is worth mentioning that a lot of these processes are still very little is understood about them in totality. I've very much glazed over a lot of stuff, especially with these SNARE complexes here. They are really, really complicated protein structures. And so there is still a lot about them that we do not understand, a lot that we've only very recently discovered. So, this is very much a simplification. And as always in my videos, I will include the disclaimer. I'm not an expert in this field. I'm simply learning about this content myself. And making these videos is a way to help me understand it and hopefully help you guys understand it a bit better as well. And so speaking of which, if you guys did enjoy the video, then please do consider giving a like and a comment on the channel. This is a brand new channel. This is still one of my first few videos I've made on this channel. So it'd be great to get some traction behind some of this content.